Okay, good afternoon, everybody. Our viewers at home. This is a special edition that has to do with genetics. And on genetics, we are going to focus on the examination int. Yes, focusing on the examination is very, very important for us in genetics so that we'll be able to solve some questions that might prove to be so difficult. Don't forget that we started this journey of genetics and engineering some weeks ago using the platform of Omega Bio-Edutainment. And all this while, we have been so glad that we have been having a lot of subscribers, comments, and feedback from our teaming audience. Now, examination is very much around us. So what should we do and what have we been able to learn all this while? And going through our educational videos, what are those things, those lectures that are definitely dedicated to genetics? Yes, I think we have more than 15 videos that were being dedicated on Omega Bio-Edutainment to genetics. Now, let's first of all just go straight to some of these major points. Number one, we started trying to introduce to us some genetical terms because every language or every subject they have their own language by which when you want to do well in that subject you should be able to be familiar with some of these key terms and terminologies in genetics. Genetics itself actually means gene, from gene, the word gene, thing that is the beginning or genesis, the beginning of gene heredity, that is the source of gene. So in one of the videos, I was able to clarify to us what we actually mean by gene and what is gene. So we introduce ourselves to what do we actually mean by gene. Gene in our modern day genetics is the unit of inheritance as it was proposed and propounded by the father of genetics, which has to do with Mendel. That time, Mendel was the first that introduced to us that certain characters or certain traits or certain units of heredity can be passed from parent to their offspring. And immediately after he must have left, died, we have a lot of genetics that took after his work and were able to confirm, revalidate, and were able to come up with different things. So in the modern day, we just discovered that a gene is just a segment on DNA, segment of nucleotide on DNA, and the DNA happens to be the hereditary unit in all organisms except in viruses, where we have some viruses using a different molecule entirely, which is the ribosomal RNA. Which is ribose nucleotide that is a RNA. That is ribonucleotide. So nucleic acid. So that is in viruses. After that, but what we are actually very much interested in this course that we have introduced us to a lot of terms and terminologies. We look at gene, we look at the DNA. We also talk about what do we mean by traits. I've mentioned it. That is a unit, that is a character, the unit that must be passed from parent to the offspring. We are also able to consider what do we mean by genotype, what do we mean if we talk about phenotype, what is Mendelian ratio, what do we understand by Mendelian ratio, Punnett square. This is very important, Punnett square. Okay, we chromosome, we made mention of chromosome. What do we mean by chromosome? You need to get yourself familiar with that. And we have F1 and F2. Okay, this is first filial generation and second filial generation. Then what do we mean by gametes? So we'll come across this. Then at a point in time in the videos, we also went as far as considering some symbolic representation of some, genetic terms and terminology, where we consider pedigree and so on and so forth like that. Okay, so that is going to cover us with, you need to get yourself again, go and revalidate your understanding of genetic terms, because that is the introduction. And don't forget that I said in the introduction, I've also said that Mendel is being regarded as the father of genetics. Why is he being regarded as father of genetics? Because he did a lot of work on that. So that one will introduce us to what do we mean by dominance, what is recessive, okay, what apart from that, we will also come across co-dominance, leader genes. These are still part of leader genes. These are still part of ABO block grouping system, and so on and so forth. So you will do well in any genetics examination by getting yourself too used to all these genetic terms and terminology. Time is going to fail us, and that is the reason why we won't be able to start going into genetic terms. So let's just move straight to the second aspect of this exam focus, okay, and that has to do with Mendel's laws. So verification under normal circumstance, you need to know that at this point in time, Mendel's law or Mendelian laws or Mendelian inheritance, there are basically three types, and you should be able to cite examples in each case, hypothetical example, that will be able to tell us that you really understand what is Mendel's law. Don't forget that we said this is the man that is being regarded as the father or the foundation of genetics. So he is referred, is up to today. So, and he was able to come up because he used a plant called Thymium sativum. That is the garden pea plant that he made use of his experiment. And there were a lot of reasons why he chose to make use of this garden pea plant. And basically in some of the videos, I was able to point out one of the reasons. I mean, some of the reasons, I think we had up to 4 reasons why Mendel chose garden pea as his own instrument for the verification and validation of Mendel's law. So the first law is called the law of monohybrid inheritance. So that is how one character can be shared, that is, or can be the pattern at which one character mono, because it is monohybrid inheritance. So if you are picking the height, that means you want to know how it is going to be from the parent to the offspring. Don't forget that we said genetics or what you call heredity. Genetics is the study of heredity and variation. Heredity and variation. If we mention what is called variation, grammatically, you might think that variation has to do with differences, but in genetics, variation is actually referring to the similarities as well as differences. So the study of variation is called genetics. Then the other aspect of it, which has to do with heredity. Heredity is the pattern of inheritance, of how gene can be passed from their parent to the offspring. So, and that's why we have discontinuous variation as well as a continuous variation, and we have a lot of traits or a lot of characters in different this. So don't forget that in some of the videos I also cited examples of what are those traits or characters. So by the time Mendel actually made use of these plants, he was able to focus on some, not all the characters that are hereditary. So, and that means part of what you should know, that not all characters that are hereditary. If you pick up an organism now, and when you want to verify the pattern of inheritance in that kind of organism, you want to know, and you should be able to know that not all characters, not all traits can be regarded as a hereditary, so to say. Okay, so now monohybrid inheritance, that is, you are picking one character at a point at a time. So, and this one is also called the law of germinal unit or law of segregation. Law of germinal units, okay, and what we call law of segregation. So we are still referring to first law anytime you come across that. So if you want to demonstrate, you want to explain the law of monohybrid inheritance, if you come across, it might not be regarded as a monohybrid inheritance here. We call it verified law of germinal units or law of segregation. So you should be familiar with, and that is, you are picking just one character at the same time. So if you have a plant like this, which is a thorn, you are crossing it with another plant that is short. So from what it is known from this, it is telling us that this is going to be capital letter, and this is going to be small letter. Allele, you have to be you have to also get yourself acquainted with the word allele, and that each character, the traits of any organism, the trait you see in any organism is a product of half from the mother and half from the father. And that's why you say that a trait will always exist in pairs. So monohybrid inheritance, when you have just one character, like the height of that plant. So, and when you do the crossing now, so I told you that you should make sure that the cross sign is quite distinct or quite different from the cross sign that you have in mathematics. So it's going to be like this. And when you do that, so that is telling you that it is going to be C. You are crossing it with this, okay, which you must know. Okay, now having said that, I think the next thing is to be able to look at this. Okay, you should be able to know, and this pattern of inheritance is what is going to follow the first law of monohybrid inheritance. Okay, going to follow that. Please, I want you to take note and think of any hypothetical example. Think of any hypothetical example that can be used to verify this. I have just picked the height of a plant. You can also make choice of another one where you are going to have a representation of all this. Okay, so I will be right back. So just like I said earlier on, we started with law of monohybrid inheritance, where we have only one character being used as a representative. So if you have examples, stroke case studies, example, okay. So let's say we have a flower. Okay, when we have a plant with red flower, okay, cross with a green flower. So we have to verify the pattern of crossing, stroke Mendelian, stroke verification of Mendelian inheritance. So I'm just going to give you an hint on this. So just say, let the red flower be represented by capital letter R. So that is going to be capital letter R, especially if in this case we are expecting it to be in a homozygous. So the allele for that is going to be the allele, which is green, is going to be represented by small letter R, and this one is going to be in recessive, so going to be RR. So you are just to verify the pattern of crossing. That is, what you are going to do is you are crossing R with this. Okay, don't forget that all this is going to stand as a unit, germinal unit. Okay, this one also like this, you are crossing it with this, so that by the time you do this is going to be your first filial generation. And what first filial generation is going to be is that all will be red. It's going to be red, and if it is red, it's going to be in heterozygous condition, that is, which is going to be capital letter R, small letter R. Now, to be able to get to the second filial generation, you are going to cross the product of first filial generation. You are crossing it with another one, that is, you are crossing heterozygous. You are crossing it with another heterozygous condition, so that the unit of this is going to be capital letter R, small letter R, crossing it with capital letter R, small letter R. And by the time you do the crossing, you are going to have 3:1. This is the exact, you have validated the Mendelian principle when you have 3:1. That is very, very important. That's very, very important. Okay, so that is what we are trying to do, that you should be able to have a kind of a situation whereby you can verify the Mendel's first law, or what is called the pattern of crossing in Mendelian inheritance. The Mendelian inheritance is going to be that the first filial generation all will be heterozygously red, and the second filial generation, since we are going to cross the results of first filial generation together, is going to be capital letter R, small letter R, with capital letter R, small letter R. And by the time you do this crossing, you are going to obtain what is going to be 3:1. If at the end of the day you now have a different ratio a different phenotypic ratio which is not 3:1, that should tell you that such a plant that is being assumed to be is not according to the Mendel's law. So if you are going to cite any example, make sure you cite the example that the second filial generation, the first filial generation, all will be heterozygous, and the second filial generation, they are going to have 3:1. That is very, very important. That's very, very important, and I think you should be able to cite example. This is you can cite it, think of any example, and the example you are going to think should follow that pattern, and that is telling you that according to the first law of Mendel that stated, or on the law of germinal unit, or what we can call the law of monohybrid inheritance, will follow this pattern, and that is very, very important for you. Okay, so please take time to be able to cite this example. Okay. Okay, just like I said, that you should be able to. The other aspect is a non-Mendelian principle. That means they will not be able to conform with the assumption of Mendel. The law of dominance is not going to be applicable in such conditions. And there are four. I think there are four different. No, there are about eight. You should be able to tell us what happens in all the eight. Tell us what happens, and you should be able to give hypothetical examples or case studies in each case. Remember, in that video I was able to give you some of this, in fact more than eight. Then don't forget pedigree and consanguinity. Okay, you need to know more the practical application of pedigree, so you can think of any case where you can make use of pedigree. If you have to write notes on it, or discuss it in details, you'll be able to tell us about that. The practical examples, the application or applicability of it, and so on and so forth. Okay. Now, after consanguinity and pedigree, you'll be able to talk about mutation. You're able to talk about mutation, what has to do with the molecular implication of mutation. We'll be able to talk about genetic disorders, what are the clinical implications, and how can it be diagnosed, as well as in case of treatment, what are the how can it be the clinical manifestation. Okay, you should be able to talk about that. I've mentioned like four. Now, four broad topic. Mendelian principle is going to be one. You should be able to anything that has to do with Mendel, details of it, be able to write a comprehensive note on it. The same thing with non-Mendelian principle, be able to write comprehensive notes. So even if you do not know anything, you should be able to know and write extensively on it. Then I said about pedigree and consanguinity, be able to write in detailed essay, comprehensive essay, hypothetical examples in each case. Then apart from that, you should be able to tell us about DNA composition, the structure, the composition, the base pairing role, what has to do with genetic code or genetic codon, how can you mime and how can you remember all this. That is another one. Okay, so then again, on genetic disorders, you should be able to know genetic disorders, what are the clinical implication of a genetic disorders, what happens when we talk of trisomy, chromosome 21, recessive chromosome 21, cradle charts, some basic examples of clinical case in each case. Genetic code, you should be able to know that. So having done that, I'm very, very sure that you should be able to come out in a well-defined color of success by the time you are able to prepare ahead and read all the thing. I want you to think of any practical examples, application of Mendelian principle, non-Mendelian principles, consanguinity. You should be able to talk extensively and discuss them in detail. Don't forget that whatever you are going to do is going to be in form of over 60. That's going to be over 60. And the grading system in this course is such that 20 marks will be devoted for for quiz, which is a CBT, that is a computer based. Some of you are confused when I said computer based. Even what you can do on phone is also computer based. Okay, the next 20 is going to be divided into two. The attendance is going to play a critical role in that, as well as a very, very important role, as well as the assignments. If you are given any assignments, I think there were times where I gave you assignments, stroke task, especially the DIY, the do it yourself. So if you do not submit your DIY, that means you are playing with your 20 marks. So even if you have 100% attendance, if you do not submit your DIY, your attendance is going to be 0. So then the last 60 is going to be in the exam. And that exam, I'm very, very sure that it is either you are given 7 questions out of which you have to answer 5, or you are given 6 questions out of which you have to answer 4. So if you are given that, that means even if each of these attracts 25 marks, okay, to make it 100, it will still be reconverted into 60% again. So that's the more reason why that it is expected that if you want to pass this course, you need to be working for something like 60%. So that means in that 100%, if it is over 100, you should struggle to score more than 70%. You are going to have a very good mark in the exam. So, once you have the strategy, once you have the tactics, facing any opponent, which is going to be your exam, will not be a problem for you. So, once again, I want to really wish you for your listening attention to the lecture series in Omega Bio-Edutainment, and I want to really seize this opportunity to stop here today. We are still going to meet in the other time on another course, especially in animal breeding and some other things that might come.